Macroevolution. Evolutionary change above the species level, including the origin of a new group of organisms or a shift in the broad pattern of evolutionary change over a long period of time. Protobiont. A collection of abiotically produced molecules surrounded by a membrane or membrane-like structure. Ribozyme. An RNA molecule that functions as an enzyme, catalyzing reactions during RNA splicing. Radiometric dating. A method for determining the absolute ages of rocks and fossils, based on the half-life of radioactive isotopes. Half-life. The amount of time it takes for 50% of a parent isotope to decay. Geologic record. The division of Earth's history in two time periods, grouped into three eons: Archean, Proterozoic, and Phanerozoic, and further subdivided into eras, periods, and epochs. Stromatolite. Layered rock that results from the activities of prokaryotes that bind thin films of sediment together. Endosymbiosis. A process in which a unicellular organism engulfs another cell, which lives within the host cell and ultimately becomes an organelle in the host cell. Serial endosymbiosis. A hypothesis for the origin of eukaryotes consisting of a sequence of endosymbiotic events in which mitochondria, chloroplasts, and perhaps other cellular structures were derived from small prokaryotes that had been engulfed by larger cells. Cambrian explosion. A relatively brief time in geologic history, about 535 to 525 MYA, when large, hard-bodied forms of animals with most of the major body plans known today appeared in the fossil record. Continental drift. The slow movement of tectonic plates across the Earth's surface. Pangaea. The supercontinent that formed near the end of the Paleozoic era, when plate movements brought all the land masses of Earth together. Mass extinction. A period of time when global environmental changes lead to the elimination of a large number of species throughout Earth. Adaptive radiation. A period of evolutionary change in which groups of organisms form many new species whose adaptations allow them to fill vacant ecological roles in their communities. Heterochrony. Evolutionary change in the timing or rate of an organism's development. Metamorphosis. the retention in an adult organism of the juvenile features of its evolutionary ancestors. Homeotic gene. A type of master regulatory gene that controls the placement and spatial organization of body parts in animals, plants, and fungi by controlling the developmental fate of groups of cells. Protocell. An abiotic precursor of a living cell that had a membrane-like structure and that maintained an internal chemistry different from that of its surroundings. Oparin Haldane hypothesis states that conditions on early Earth, in particular, the reducing atmosphere, lightning, and intense ultraviolet radiation, favored the synthesis of organic compounds. Stanley Miller, an American chemist and biologist, 1930 to 2007, who is known for his studies into the origin of life, particularly the Miller-Urey soup experiment in 1953, which demonstrated that organic compounds can be created by fairly simple chemical processes from inorganic substances. Abiotic synthesis. The building of biological molecules from inorganic precursors. Montmorillonite. A soft mineral clay produced by the weathering of volcanic ash that greatly increases the rate of vesicle self-assembly. Vesicles. Fluid-filled compartments bounded by a membrane-like structure, can exhibit certain properties of life, including simple reproduction and metabolism, as well as the maintenance of an internal chemical environment different from that of their surroundings. RNA world. A hypothetical period in the evolution of life when small, self-replicating RNA molecules served as rudimentary genes and the sole catalytic molecules. Placoderm. A group of heavily armored jawed fishes of the Silurian and Devonian periods. Tiktaalik. A genus of extinct Sarcopterygian, lobe-finned fish from the late Devonian period, with many features akin to those of tetrapods, four-legged animals. Burgess Shale. A fossil formation in the Canadian Rockies that contains Cambrian soft-bodied organisms as well as organisms with hard parts. Tetrapods. A vertebrate with four limbs, including mammals, amphibians, birds, and reptiles. Cenozoic. A type of theropside, advanced mammal-like reptile, the ancestors of mammals. Theropside. A reptile order that had some small members that evolved into little nocturnal mammals in the Triassic period, had large dentary bones, long faces, and large canines, the first examples of specialized teeth. synapside. An extinct reptile having a single pair of lateral temporal openings, fenestrae, in the skull, had multiple bones in the lower jaw and single pointed teeth. Paleozoic. An era occurring between 542 to 251 million years ago, characterized by the advent of fish, insects, and reptiles, includes the Cambrian, Ordovician, Silurian, Devonian, Carboniferous, and Permian periods. Mesozoic. An era with the Triassic, Jurassic and Cretaceous periods from 251 to 65.5 million years ago, marked by dinosaurs, gymnosperms and angiosperms, as well as the mass extinction at the end of the Cretaceous period. Cenozoic. An era with the Paleogene, Neogene, and Quaternary periods, 65.5 million years ago, to present major radiation of mammals, birds, and pollinating insects. Angiosperm dominance increases, origin of many primate groups, human evolution. Ediacaran. A period with the earliest generally accepted animal fossils, soft-bodied invertebrates, dating from about 635 to 542 million years ago. Algae were diverse. Cambrian. A period in the Paleozoic era with a burst of evolutionary origins when most of the major body plans of animals appeared in a relatively brief time in geologic history, 542 to 488 million years ago. Ordovician. A period in the Paleozoic era when marine algae were abundant, colonization of land by diverse fungi, plants, and animals, 488 to 444 million years ago. Silurian. A period in the Paleozoic era where early vascular plants diversified, 444 to 416 million years ago. Devonian. A period in the Paleozoic era when bony fishes diversified, first tetrapods and insects appear, 416 to 359 million years ago. Carboniferous. A period in the Paleozoic era when extensive forests of vascular plants form, first seed plants appear, origin of reptiles, amphibians dominant, 359 to 299 million years ago. Permian. A period in the Paleozoic era marked by the radiation of reptiles, the origin of most present-day groups of insects, the extinction of many marine and terrestrial organisms at the end of the period, possibly triggered by volcanic activity and a drop of oxygen in oceans, 299 to 251 million years ago. Triassic. A period in the Mesozoic era where cone-bearing plants, gymnosperms, dominate the landscape. Dinosaurs evolve and radiate, origin of mammals, 251 to 199 million years ago. Jurassic. A period in the Mesozoic era where gymnosperms continued as the dominant plants, dinosaurs abundant, and reverse 199 to 145 million years ago. Cretaceous, a period in the Mesozoic era where flowering plants, angiosperms, appear and diversify. Many groups of organisms, including most dinosaurs, become extinct at end of period 145 to 65.5 million years ago. Oxygen revolution, the time span, from about 2 billion to 400 million years ago, during which photosynthetic autotrophs changed the composition of Earth's atmosphere to its current oxygen-rich mixture. Endosymbiont theory states that mitochondria and plastids, including chloroplasts, originated as prokaryotic cells, engulfed by an ancestral eukaryotic cell. The engulfed cell and its host cell then evolved into a single organism. Snowball Earth hypothesis states that glaciers covered the planet's land masses from pole to pole 750 to 570 million years ago, confining life to very limited areas near deep-sea vents and hot springs or to equatorial regions of the oceans that lacked ice cover. Plate tectonics, the theory that the continents are part of great areas of Earth's crust that float on the hot, underlying portion of the mantle. Movements in the mantle cause the continents to move slowly over. Hox genes, a series of genes that controls the differentiation of cells and tissues in an embryo.